Hey guys, so today we're gonna be doing respiration again. So this is gonna be the final part of the topic. So we're gonna finish the chapter by today. Now let's start. We're gonna start with adaptations in rice plant. Okay, so I'm just gonna read this page out loud for you guys first. Then I'm gonna explain. Oxygen supply isn't usually enough to supply all the energy the cells need by aerobic respiration as plant is submerged in water. Gases diffuse much more slowly in the water, so some rice plants grow upwards so that the top of their leaves are held above water, allowing O2 and CO2 to be exchanged through the stomata on the leaves. Now, rice plants are semi-aquatic plants. The roots and some portion of the shoots, they're like submerged in water, and diffusion of gases is like really slow in water, right? So the cells in the rice plants, they cannot get adequate amount of oxygen. So that's why they got something called parenchymas. What are parenchymas? Stems of the rice plants contain loosely packed cells um, that form parenchyma tissue. So parenchymas are tissues that are large, and they got like large air spaces which allow efficient gas exchange. So what happens is that gases from the shoot goes into the parenchyma, then to the plant, right? So I'm gonna discuss the function of parenchyma now. Main function, gas exchange. It allows oxygen to move from above water parts like leaves down to the submerged tissues like roots. This is crucial in waterlogged or flooded conditions where oxygen is limited. Okay. Why this matters? In waterlogged soils, root cannot get enough oxygen for respiration. Parenchyma provides an internal air channel, helping roots survive by supplying oxygen and removing gases like CO2. So it doesn't only take in O2, it also releases waste like carbon dioxide, right? So where it's common in aquatic or semi-aquatic plants. Okay, this is not needed. Extra function, buoyancy, the air spaces can help some plants float. You don't need this either, but I thought it's interesting, so I just put it there. Now, first adaptation is that the stem growth is accelerated, okay? And second adaptation is they got parenchymas. They got parenchymas. And now the final adaptation that's in our syllabus. So still, root cells of rice plant use alcoholic fermentation sometimes. So alcoholic fermentation, yeah. So this produces ethanol. And this is an anaerobic process. And excess buildup of ethanol can be toxic to the tissues. That's why rice plants are generally much more tolerant to ethanol. Also, they produce alcohol dehydrogenase in larger amounts so that the ethanol can be broken down, right? So the third adaptation is more tolerance to ethanol. I'm just gonna write it OH, and we got alcohol dehydrogenase. So the alcohol dehydrogenase is gonna break down ethanol, allowing plants to actively grow even when O2 is scarce, using ATP produced by alcoholic fermentation. Now, excuse me. No, alcoholic fermentation. This is an anaerobic process. This produces 2 moles of ATP, right? So the plant is gonna use the 2 moles of ATP to grow I mean, it's gonna not 2 moles, literally like multiple cycles of anaerobic respiration is gonna take place. So this is it for the adaptation and rice plants part. We're gonna move. We're gonna move to something called RQ, or respiratory quotient. So what is an RQ value and why do we need it? So RQ is basically the ratio of CO2 produced over the O2 used. Okay, is the ratio of CO2 produced and oxygen used. Now it tells you what type of substrate is being used. Now, what is a substrate or respiratory substrate? Organic molecules that can be broken down to generate ATP. The type of respiratory substrates are carbohydrates, lipids, proteins, and carbohydrates, lipids, and proteins. So these are your respiratory substrate. We can use something called a respirometer to measure the RQ values, right? Here are like some RQ values for the type of substrate being used. For glucose, the substrate is the RQ is 1. You have to memorize this, by the way. For lipids, the RQ is 0.7. The accepted range is 0.7 to 0.8. This is only aerobic. For proteins, RQ is 0.9, only aerobic. Now, I want you guys to know something, is that when anaerobic respiration takes place, the RQ is going to be greater than 1. Why? Because there's going to be no oxygen used. So when something gets divided by 0, it's basically infinite. So RQ greater than 1 is going to indicate anaerobic respiration. All right. Now let's take a look into, okay, what are these? Okay, you don't need this, not this either. Apparatus used to measure RQ is a respirometer. Now we're gonna take a look. Okay, before looking into a respirometer, we're gonna look at the energy values, right? Energy values: amount of energy released per gram of a respiratory substrate. Carbohydrates, 15.8 kilojoule like per gram of carbohydrates. 1 gram of protein, 17, and lipids, it's like almost 40. So you may be asking, why is it so high? That's because lipids, if you remember from your AS, lipids are long chain hydrocarbons. So there are more CH bonds present, meaning there are more hydrogen atoms, and hydrogen atoms split to form protons and electrons. And these make your NADH, reduced NAD plus, or FADH2. The NADH and FADH2, they release their electrons in the ETC, and in the cristae of the mitochondria, chemiosmosis takes place. Basically, oxidative phosphorylation takes place, and more NADH and FADH2, meaning more oxidative phosphorylation, and oxidative phosphorylation creates ATP. So more oxidative phosphorylation equals more ATP. Simple math. Anyways, we're gonna take a look into respirometer, right? So this is called a simple respirometer. Okay, and let's see, what do we got here? We got a tiny bug, a small organism. We got gauze. Why do we need gauze? Because we got soda lime here. Soda lime is potassium hydroxide, calcium hydroxide, blah blah blah. So soda lime, if soda lime comes in contact with the organism, it can damage the organism, and we don't want that. So we need a barrier. So gauze acts as a barrier. We got and also we got to make sure that the tube is airtight. So we use a bung, and we use a syringe. This is basically to repressurize the tube. Okay. We also got a capillary tube going out from the test tube. We got a scale here, and then we got colored liquid, a drop of dye or anything really, a drop of colored liquid. Now, let's discuss how all of these work together, right? Now, the respiring organism is gonna use oxygen and produce carbon dioxide. If you look closely, the ratio is 1:1, meaning uh, the pressure is gonna be unaffected. Like the tube, that there won't be any pressure changes in the tube, right? So we need to create pressure change to analyze how much oxygen is being used. So to create pressure changes, we use soda lime. Soda lime, it acts as an absorbent. It's going to absorb the carbon dioxide. And this is going to create a pressure difference between the capillary tube and the test tube. Now, the pressure inside the test tube is going to decrease as soda lime absorbs the carbon dioxide that is being produced by the organism. And by the way, we can use maggots, we can use lice, germinating seeds, basically any small living respiring organism. Now, this pressure change is going to cause the oxygen present in the capillary tube to move in, and as the oxygen moves in, the colored liquid is going to move along. It's going to move along, like it's going to move too. And we can study the rate of oxygen intake by timing how much oxygen how much oxygen is being moved. So it's like this. The formula goes like this. It's like volume of O2 by time. So also one thing, the diameter of this capillary tube should be known, okay? Because we need to find the volume, right? So diameter should be known. Now let's say I wanna see how much oxygen is being used by this organism right here in, let's say, 10 minutes. Okay, so first I'm gonna find first I need to write an example value. So let's say in 10 minutes the colored liquid, it moves let's say 15 millimeters 15 millimeters towards the test tube. Okay, now we're gonna find the rate of oxygen intake. Okay, so time was 10, and to find the volume, we're gonna do is first find the area of the what is it, the capillary tube. So it's gonna be pi r square. Let's take the radius as 0.5 r square into this is also in millimeters, by the way, and this is gonna be 15. So this is gonna give you the volume 15. Let me just do the math. Give me a sec. 0.5 squared to 15 by 10. It gives me 17 millimeter cube minute. Yes, this is correct. So this is my rate of oxygen intake. Okay, now we're gonna discuss how it's done. So usually in exams, these come as like an 8-marker questions, right? So I'm gonna discuss wait, give me a sec. Right. So I'm gonna discuss how it's carried out. So the apparatus needed is simple respirometer, capillary tube, colored liquid, organism, your absorbent, we need water baths at different temperatures, right? We want to see like how temperature affects the rate of oxygen consumption. So we're gonna need water baths at different temperatures. We're gonna need stopwatch to like time it, and the ruler is basically the scale. So method: place organism in the respirometer at sodium hydroxide to absorb CO2. Seal the apparatus airtight. Place respirometer in a water bath at a known temp. Allow time to equilibrate. So basically, give it time, like for the test tubes to acclimatize. Basically, the temperature of the water bath and temperature of the test tube should be equal, and it takes time for them to be equal. So you're gonna have to give them time. Record movement of colored liquid over time. Repeat at different temps, and then calculate rate of respiration as distance moved by time. Okay. Now, what is being measured? CO2 is absorbed only. O2 uptake causes volume decrease. Movement of liquid equals to rate of oxygen consumption, which is equals to rate of respiration. Now, the variables. We are changing the temp, and changes in the temp directly changes the rate of respiration, right? So the independent variable is going to be temp, and the dependent variable is going to be rate of respiration. And these are control variables, like they should be constant. You cannot change them. You should not change them. So the mass of the organism should be constant. The species should be constant. Volume of air, time travel, pH, blah blah blah. They should be constant. Okay. So conclusion, we can draw a conclusion that rate of respiration rate increases with temperature due to increased enzyme activity up to an optimum, after which it decreases due to enzyme denaturation. Okay. Key precautions: ensure airtight system, or else the pressure is going to be you're not going to get the appropriate pressure. Allow temperature to equilibrate. Now you're going to have to give it time, so that the temperature of the water but then temperature of the test tube is the same. Use replicates and calculate mean. So you're gonna take multiple, like you're not just gonna do one experiment, you're gonna do multiple experiment, and then you're gonna take the mean. Also take control. This is for validity. Okay, use control for validity. Use glass beads as example. So that would be it for his pyrometer. Now then we got something called redox and indicators. Now in our syllabus we got DCPIP and methylene blue. So DCPIP and methylene blue, they are both blue in color, okay? And they can do what is they can accept protons. So it can produce protons, and after it accepts proton, it changes color. So the blue color changes, it fades, and it decolorizes. So it's gonna be like this. The observation will be blue to colorless. Okay. So how can this help us understand the rate? So the faster the color change takes place, the faster the rate is, the higher the rate is. Okay. So we got a test tube with yeast cells, and then we add any either of the indicators, DCPIP or methylene blue, and then we time it. We see how much time it takes for the indicator to change its color. So we can vary the concentration of the yeast cell, we can vary the temp, blah blah blah, to study the rate of respiration, the effect on the rate of respiration, right? So glucose, it's gonna get dehydrogenated to pyruvate, and then it's gonna release H plus protons, right? So normally the H plus gets gained by NAD plus to form reduced NAD, which is NADH. But in this case, the the protons are going to be accepted by DCPIP or methylene blue. Okay, so yeah, that's it. This is the normal equation. Effect on temperature: 10C 100 seconds to change color, 20C 70 seconds means faster rate of respiration. Redox indicators change color when they get oxidized or reduced. Okay, methylene blue origin color is blue. Add redox indicator. These are yeast cells. They decolorize. During respiration, dehydrogenation takes place in glycolysis, link reaction, Krebs cycle. The H plus released as a result of that can be accepted by DCPIP or and methylene blue. This results in the reduction of DCPIP or methylene blue, causing them to decolorize. Faster the rate of respiration, more hydrogens are released per unit time, and faster the dyes are reduced, faster they're decolorized. The rate of change from blue to colorless is a measure of the rate of respiration. What else do we got? Um, okay, this is not really related. Lipids have more. Okay, this is not related. Okay. All right, guys. So we're done with respiration. So if you guys have any suggestions, you can comment below and let me know. I'm gonna do photosynthesis next. So yeah, hopefully see you in the next video. Bye, guys. Take care.